Bismillahirrahmanirrahim. We start another video. This is the second video on March 2023, paper 4-2, and we'll probably be discussing question 3, 4, and 5. Now, starting with question number 3, salmon can be genetically modified GM to produce increased quantities of growth hormone, which is a protein. GM salmon modified in this way have a faster growth rate and reach their maximum body mass at a younger age than non-GM salmon. Within any population of salmon, there is variation in body mass. This is an example of continuous variation. Explain what is meant by continuous variation and how it can be caused. So basically, what is continuous variation? Is phenotypes that change gradually over a range of values. Or you could have said that phenotypes that do not fall into distinct groups or classes, because there's a discontinuous variation, like blood group is discontinuous. There are only 4 blood groups. So there are 4 distinct categories. But continuous variation is phenotypes that change gradually over a range of values. So the mass is very variable. They can be mass of 10 gram, 20 gram, 30, 40, 50, up to maybe even 500 grams, or maybe even more than that. And the intermediates connect the extremes, and there's a normal distribution. But besides that, it says explain what is meant and how it can be caused. Because it is controlled, it's polygenic, means it is controlled by many genes. And different genes have an additive effect. If you have this gene, and if you have this gene and this gene, then they will all add up and result in a certain mass of the fish. Environmental factors contribute to the variation, just like the height of a person will be dependent on the diet if he gets in his early teens. And of course, the genes which are present are there, but then of course, if he doesn't get the right food, just like a plant not getting enough light will not grow tall, or a plant not getting enough nitrates from the soil will then result in stunted growth. So continuous variation is affected by the environment. So environmental factors contribute to the variation. Now this is 3 marks. So there will be 2 marks for what is meant by continuous variation, and then any 1 or 2 points for how can it be caused. Do not fall into distinct groups, like just like I told you, discontinuous variation is in blood groups, and there are 4 distinct categories, or you can say if you convert the positive and negative, so 8 distinct categories. Then intermediates connect the extreme. So there is a very large intermediates. They can be varying between, say, 1 kilogram to 10 kilogram, something like this. So intermediates connect the extremes. There's normal distribution, and then the fact that what causes them is polygenic, controlled by many genes, and then different genes have an additive effect. If you have this allele, this allele, this allele, this will contribute to it, so it will have an additive effect. Environmental factors contribute to variation, and I just gave you an example of if they don't get enough diet, if they don't get enough calcium, if they don't get enough amino acids and glucose, well, they won't grow that fast, the fish. But we can talk about plants as well. If they don't get enough light and they do not get nitrates from the soil, well, they will not grow tall. Then coming to the B part of the question, scientists investigated whether injection of very young non-GM salmon with recombinant growth hormone could cause an increase in the growth rate of the salmon. The scientists used 2 groups of non-GM salmon, a control group of salmon that were not injected with recombinant growth hormone, and an experimental group of salmon that were injected with 1.0 micrograms of recombinant growth hormone at the start of the experiment and once a week for the next 6 weeks. The mean body mass of the salmon in the 2 groups at the start of the experiment was the same, 5.3 grams. After 6 weeks, the body mass of every salmon was measured again. The results are summarized in table 3.1. So we've got all these figures here. So number of GM salmon, body mass, range, mean, standard deviation. A student decided that a t-test should be performed on the results shown in table 3.1. Calculate the value of t for the result shown in table 3.1 using the formula for the t-test. So I've told you many times the formula is always given to you, but how you have to apply then. Give your answer to 2 decimal places. Show your working. Now, as you look at the formula, it was very easy. You see the means, x1 mean. So this was 7.7 here and 9.4 here. So I put that here, and 7.7 minus 9.4 is 1.7, right? And then you have to put the standard deviation 1 over n1 and standard deviation 2 whole square over n2. So you can see here standard deviation here was given 0.4 and here it was given 1.1, and then n1 was 28 and then it was 27. So you had all these figures, you just had to put all this in. Please try to do it on your own first and then figure it out, and then you have to do the calculation. So t is equal to 7.56. Now as the question proceeds, the critical value of p is 0.05. At probability 0.05 for this data is 2.01. Now look at your figure. So the student used the results in table 3.1, the t-test, to conclude that the injections of recombinant growth hormone cause an increase in the growth rate of the non-GM salmon. Comment on to the extent to which the conclusion made by the student can be supported. You see the t-value 7.56 is greater than 2.01, or you can say it's greater than the critical value, because 2.01 is the critical value. This is called the critical value. I've talked about this in so many of my videos. So t-test may not be valid due to the unequal variances. Then overlap in data range means not effective for all salmon. Correlation does not prove a causation. To this is only 5% probability, so may not actually be different. Only a single investigation or experiment or a set of results so needs to be repeated so that we can get confidence in the conclusions. And both groups show an increase in body mass from the start of the experiment. So this is how you had to sort of tell me that if because the critical value is the t-test is more than the critical value, so then that means the conclusions are not sort of very substantially supported. So t-value 7.56 is greater than 2.01. That was an easy one mark to get. T-test may not be valid due to unequal standard deviation, and correlation does not prove causation. Only a single investigation needs to be repeated so that we have more confidence in the conclusion. And both groups show an increase in body mass from the start of the experiment. So any 3 points out of these, you could have got your 3 out of 3. Last part, suggest, which is always very difficult, suggest one advantage other than cost of farming GM salmon that produced increased quantities of growth hormone instead of farming non-GM salmon that are injected with the recombinant growth hormone each week. You see, GM salmon may grow even faster. Idea of, there's something less labor intensive, and growth hormone concentrations are more consistent, is continuously produced. You could say less stressful on the GM salmon, or risk of infection with injections. You could also say that. Approval for GM salmon obtained in some countries, but not for regular injections. So one advantage other than the cost, you could have said, the fact that GM salmon may grow even faster. And well, they reach the size by which they can be sold in the market. So I've just given 1 point because there's one mark for that. Of course, there are many points, but GM salmon may grow even faster. But the main thing is approval for GM salmon is obtained in some countries. So it is a little difficult to have this genetically modified animals, and people resist these. I mean, I'm very much against this as well, and I don't believe in this. Then coming to question number 4, array comparative genome hybridization (ACGH) is a technique involving the use of a microarray to analyze a genome or sections of a genome. Outline the steps required to prepare the genome of an individual so that the genome is ready for analysis using a microarray chip. Extract DNA from cells. Denature into single-stranded DNA. Cut DNA into small fragments and add fluorescent tags. So that was a very basic direct question. Extract DNA from cells or tissues, denature into single-stranded DNA, which is also called ssDNA. ssDNA, single-stranded DNA, cut DNA into small fragments using restriction enzymes and add the fluorescent tags so that then we can say whether they're red, green, or yellow if they're present in both. So we do a comparative genomic analysis. Now coming to the B part of the question, DiGeorge syndrome is a dominant inherited disease in humans. DiGeorge syndrome is caused by a deletion of a large number of nucleotides from chromosome number 22. The number of nucleotides deleted varies between individuals in a range from 800,000 to 3,100,000. The largest deletions can cause the removal of up to 46 protein coding genes from the chromosome. Figure 4.1 shows the results of ACGH using a microarray specific for the section of chromosome 22 within which the DiGeorge syndrome deletion occurs. The microarray analyzed DNA from two individuals, one with DiGeorge syndrome, one who did not have DiGeorge syndrome, which would be the control DNA for comparison. In the ACGH results shown in figure 4.1, each small circle represents the results from a single probe on the microarray. The x-axis shows the position of each probe on chromosome 22. The position is shown as a distance along the chromosome in millions of nucleotides. A result close to 100% fluorescence on the y-axis means that the DNA from the individual with DiGeorge syndrome fluoresces at the same intensity as the control DNA for that probe. A result close to 50% fluorescence on the y-axis means that the DNA from the individual with DiGeorge syndrome fluoresces half as much as the control DNA for that probe. Now you will probably need to read this twice to be able to really comprehend it and then be able to further go on into the question. So here is the figure 4.1, fluorescence of DNA with an individual DiGeorge as a percentage of the fluorescence of the control DNA. Position of probe on chromosome 22, and this is in millions of nucleotides. With reference to figure 4.1, so with reference to figure 4.1, you have got to look at figure 4.1. Estimate the number of nucleotides deleted from the affected chromosome 22 in the individual with DiGeorge syndrome. Give your answer to the nearest 100,000 nucleotides. So you can see how we have calculated the 17.2, 19.8, and this is 2.6 million, which is equal to 2,600,000, not 26 lakhs. Because it says give your answer to the nearest 100,000 nucleotides. Then explain how the microarray technique works to give the results shown in figure 4.1. Now that was easy. DNA from the DiGeorge syndrome and control DNA labeled with different colored fluorescent tags. Probes on microarray have complementary sequence. Compare the fluorescence of individual with DiGeorge syndrome and the control. Then in DiGeorge syndrome, less 50% DNA binds to the probes on the microarray than the control DNA. All 100% DNA equally to probes for other parts of the genome, which is chromosome 22. DNA from DiGeorge and control DNA labeled with different colors of fluorescent dye. Probes on microarray have complementary sequence to chromosome 22. Compare the fluorescence of individuals with DiGeorge and control. You're going to compare them, like in my video I explained to you normal and cancer cells. Then in the DiGeorge, 50% of the DNA binds to probes on microarray than control DNA. So 3 marks. Basically, you have to understand if you knew microarray, you could have done this question. If you didn't know microarray, well, you couldn't have done this question. Now, the last part of this question, part 3, suggests why the phenotypes of two individuals with DiGeorge syndrome can be different. Because chromosome deletion may be of different number of nucleotides. There's a very variation amongst the number of deletion of nucleotides. So different number of genes deleted. Individuals may have inherited different alleles of the same gene. Or you can also say individuals have different environment, or you can say even in the diet, you could have been talking about the diet. And some individuals may have both chromosome 22s affected, others just one. So there was it suggests is always a very difficult question. And why different? Because the number of deletions was different. It wasn't standard that everybody with D. George had deletion of, say, 1000 nucleotides. No, it was different, it was variable. So chromosome deletions may be a different number of nucleotides, so different number of genes deleted. Individuals may have inherited different alleles of the same gene. Individuals may have different environments because we're talking of, it said, phenotype, and you know phenotype is dependent on the environment. Then some individuals may have both chromosome 22s. You see, a 22 will be a pair of chromosomes. So either one will have deletion or maybe both will have the deletion. So both chromosome 22 is affected. In some, only one is affected. So a lot of possible answers which you could come with. Question number 5. Meiosis is described as a reduction division because the number of chromosomes in the daughter cells is reduced by half. Table 5.1 describes some of the events that take place during 4 of the different stages of meiosis in an animal cell. Stage of meiosis, protophase 1, attach to centromeres and arrange homologous pairs of chromosomes at the equator of the cell. Anaphase 1 is left blank, then reform spindle in daughter cells here. So this has been left blank, this has been left blank, and then the last one is telophase 2, disassemble, and then you had to complete this. Now it says complete table 5.1 by outlining the behavior of the spindle fibers during anaphase 1. Identifying the stage of meiosis in which spindle fibers reform the spindle in daughter cells, and drawing a diagram to show telophase 2. You do not need to add labels to your diagram showing telophase 2. So this is 4 marks. And then it says explain the need for reduction division during meiosis. So very direct question, a very easy question, only 8 marks, and I'm sure everybody could do this question. So in anaphase 1, what would happen is the spindle fibers contract to pull the chromosomes towards the poles, or you could have said to the centrioles. And then it was prophase 2, reform spindle in daughter cells. And then here you have to draw 4 cells, diagram showing forming 4 daughter cells, 2 chromosomes each with a single chromatid inside a reforming nuclear envelope. And that is why this was for 4 marks, that you can see in this part was for 4 marks. Then explain the need for reduction division during meiosis. Well, that's very easy. Why would we have a reduction division? Because at fertilization, then the number is restored. So meiosis produces gametes for sexual reproduction. At fertilization, 2 gametes fuse to form a zygote. And the zygote will have chromosomes from both the gametes. And gametes must be haploid, having half the number of chromosomes. And this is will maintain the number of chromosomes. The number of chromosomes will be maintained because what will happen, the normal number of chromosomes, otherwise every generation the number would double. So if the gametes are not haploid, then when they fuse, the zygote will be tetraploid or 4n. So the need for reduction division so that the diploid number is restored in the zygote. That's a very nice statement I like. Diploid number is restored in the zygote. So meiosis produces gametes for sexual reproduction. At fertilization, 2 gametes fuse to form a zygote. Gametes must be haploid. If gametes not haploid, then zygote will be tetraploid, 4n. When haploid gametes fuse, the diploid number is restored in the zygote. A very, very nice way of expressing this. Now coming to question number 6, which is a very interesting question because it's an easy one. Respiration and photosynthesis seem to go well with all of us, and we do very well in that. Figure 6.1 is a diagram of a section through a mitochondrion. The 4 arrows A, B, C, and D show the movement of molecules and ions. Use the letter to identify all the arrows, one or more, that show active transport of protons. Active transport of protons, when was that happening? Active transport of protons was when in the ETC, In the intermembrane space, the hydrogen ions that are pumped into the intermembrane space are then going to be moving back through the ATP synthase. So active transport of protons. So which one would that be? Active transport of protons are then pumped into the, because when they pass through the ATP synthase, that's facilitated diffusion. But when they're actively transported is when the NADH gives up the hydrogen ions. So the NAD comes in here, NADH gives up the hydrogen ions. The hydrogen ions are pumped into the intermembrane space. So this is active transport. But when they are flowing like here, this would be facilitated diffusion. Hydrogen ions moving through the ATP synthase and then ATP being made. So active transport of all protons would be D, and then diffusion of carbon dioxide. What would that be? Diffusion of carbon dioxide. Diffusion of carbon dioxide, you see, you have to realize is carbon dioxide will be made in the respiring cells. Aerobic respiration takes place in the mitochondria. So it would be C. Carbon dioxide is made when? Carbon dioxide is made in the link reaction and the Krebs cycle. So that is made in the matrix. So the carbon dioxide is going to be diffusion of carbon dioxide would be C and D. Why D? Because carbon dioxide is in matrix is this place. In the matrix we have the link reaction taking place and the Krebs cycle taking place. And that is where carbon dioxide is made, so it's going to diffuse into the intermembrane space and then out. So it had to be C and D. Then outline the role of the mitochondrial matrix in respiration. Now matrix, we've just talked about that. Location of enzymes for the link reaction of the Krebs cycle. It is the site of the link reaction of the Krebs cycle, and it is the DNA and the ribosomes for the production of enzymes. Then here reduced FAD and reduced NAD are made here in the link reaction and the Krebs cycle for oxidative phosphorylation. Plus substrate link phosphorylation takes place here. So this is a recap of all what we've done in respiration. So site of the link reaction or the Krebs cycle, if I put a slash, it means either you said that or that. It's not that you have to put both of them. Then DNA and the ribosomes are present here for the production of enzymes needed for the link reaction and the Krebs cycle, and also for the electron carriers. And then production of reduced FAD or reduced NAD for oxidative phosphorylation. And then, of course, in the Krebs cycle, we also have substrate linked phosphorylation, where ATP is made in the Krebs cycle, and also it's made in the glycolysis, but that's in the cytoplasm of the cell. So we don't talk about glycolysis here. Then another very direct question, which is a very simple question: explain how a lack of oxygen affects oxidative phosphorylation. So all that which happens in the presence of oxygen, you just have to say that it will not happen. So no electrons accepted by oxygen, no electrons will flow along the electron transport chain, no hydrogen ions pumped into the intermembrane space, no chemiosmosis. Then the reduced NAD and the reduced FAD is not oxidized, so no ATP will be produced and no pyruvate will enter the mitochondrion. So all the basic things which, if oxygen is not available, neither the link reaction nor the Krebs cycle will take place, because oxygen is the final electron acceptor. So no oxygen, no electrons accepted by oxygen. No electrons accepted by oxygen. No electrons flow along the ETC. No hydrogen ions pumped into the intermembrane space. No chemiosmosis. Reduced NAD, or if you can say reduced FAD, is not oxidized. It doesn't give up its hydrogen. And no ATP produced. No pyruvate enters the mitochondria. So total 9 marks. Very easy to get full marks in this question.